Hi everyone, Professor Peter Nash from Griffith University, beautiful downtown Brisbane, reporting for Room Now at EULAR Barcelona 2025. Let's talk about the novel agents that were presented in the IL-17 class at this meeting. There were 4 of them. Just when you thought the 3 that you had was all that you needed, Chinese colleagues presented 2, avunakizumab and zaligakumab. They were IL-17A inhibitors, they were parenteral, and guess what? They worked like an IL-17 agent. They had the same safety as an IL-17A agent, and they seem to work both in PSA and AxSpA, as you would expect. The 2 other agents that are interesting, sonelokimab, if you can say these drug names, you're ahead of the pack, is interesting because it's a nanobody, 17A and 17F, binds to albumin, supposed to have superior soft tissue penetration, and the numbers are really quite good for a phase 2 study, as they usually are. They had an active comparator with adalimumab in their trial, and they were getting MDAs when they compared their doses, 60 milligrams, 120 milligrams, compared to the active comparator adalimumab. They were getting numbers of the MDA in the 50 to 60% range, which is a little higher than the normal 35-40% that we get in the phase 3 trials. Of course, the ACR70 and PASI 100 was quite significant in the higher dose in the 48% range, driven mainly by the skin response, as you'd expect, compared to the 18-19% seen with adalimumab. And again, the ACR70 was very impressive in the 40% range, and the PASI 100, you now expect 100% clearing of skin with these agents, was in the 60% range. So numbers are impressive, safety, no new signals. The MOA is supposed to be an advantage. They're going into phase 3, and we'll see if the superior numbers, as you often see in phase 2, holds up in phase 3. The other agent that was very interesting was this agent ORCA 002. Doesn't seem to have a difficult to pronounce name at this stage. Now this agent, they fiddled 4 amino acids in the hinge region, cut off the FC, pegylated it, and gave it to cynomolgus monkeys, and they extended the half-life out to 4 to 6 months. And they're aiming to use this IL-17AF monoclonal antibody to be a twice-a-year injection. If it stacks up, there weren't much in the way of clinical details, but if it stacks up efficacy and safety-wise, what an advantage to have a twice-a-year injection, unless, of course, you get something like IBD or terrible candida, when something that hangs around for 6 months might not be such a good idea. So 4 new agents, 2 that look like Me-Toos, another nanobody, and 1 long-acting agent that might be interesting. Thanks.